Everybody, welcome to this Faculty of Science information session for people who are looking to find somebody who might give them a scholarship to do research in a really cool area. I remind everybody that this session is being recorded. Everything you say or write in the chat actually gets recorded, so be aware that that's happening. If you're uncomfortable with being recorded, then now is probably the time to leave and or try not to say anything much. We've got a series of academics who are going to give you a little brief description of the kinds of projects that they're running and the fact that they have X scholarships to award to people who might be interested in the area. If you are interested in any particular area, make a note of the name of the person and then email them, organise a meeting and have a discussion about possibilities of you maybe taking up that scholarship that's available. Okay, our first presenter now, we'll move along, is Lachlan O'Neill, who's a team leader in energy data science at CSIRO, and he's talking on behalf of Professor Ricardo Campello, who's the professor in maths in data science. Thanks, Lachlan. You need to unmute, Lachlan.
How's that?
Wonderful.
Thank you very much. Thank you for the introduction, and thank you everyone for taking your time to listen to me today. So I'm from the CSIRO. So I work here as an energy data scientist, and most of my work day to day is about putting time and effort into better understanding residential energy consumption. So I actually went out of my way to try and find an academic that would partner with us, because we're starting to run into a huge problem when it comes to just the sheer volume of data that we have. So you may or may not be aware, but a lot of households have something called an interval meter installed. And what that means is for your electricity consumption, we go ahead and store what that was every half hour of the day. So you can imagine when we have millions of households in Australia, and we have half-hourly recordings of your electricity consumption. And for some households, this has been going on for up to a decade. We start getting into big data problems really, really quickly. We work regularly with a bunch of different organizations all up the supply chain. So we work right at the household consumer end with consumer advocacy groups to try and help end users of electricity. We work with retailers, we work with distribution networks, so people who actually work on the infrastructure that moves the electricity around Australia. We also work with government organisations and statutory bodies. We also talk to universities and consultancy firms. We're right in the middle of industry, trying to get to the bottom to basically try and work out how to provide better energy outcomes for Australia. So that more or less means trying to make sure that our grid is reliable, so that energy is always there for you when you need it, that it's secure, so that all the energy, all the actual components of the network aren't going to blow up because they're operating outside of their parameters, and making sure that energy is affordable as well. So this scholarship is really about trying to get on top of all those what we call low profile, so electricity consumption of hundreds of thousands of households for which we have access to data. So we do have access to a lot of this data, and specifically trying to cluster them together so that we're not thinking about hundreds of thousands of households, but rather a standard grouping of households. So Professor Ricardo Campello is the lead supervisor on this, and I'll be the co-supervisor. And one really exciting thing about this is because this is actually co-funded by CSIRO, in addition to the 28K per annum that the University of Newcastle offering, CSIRO is offering an extra 10K per annum on top of that, and an additional 5K per annum for research support funding. So if you need to go to a conference, hopefully we're allowed to fly again one day, or if you need particular software or you need particular readings or whatever, then there's funding available for that as well. So if you're interested in applying, you're welcome to reach out to myself or Ricardo. It is up on the University of Newcastle's website under Exploring Multifaceted Clustering of Complex Electricity Time Series Data to Support Data-Driven Decision-Making in the Energy Sector. But basically, if anyone's interested in this scholarship, it is a great opportunity for anyone interested in data science, anyone who gets involved in machine learning or the likes. You'll be working directly with industry, and you will be working directly with CSIRO as well. So we would love to see your application. And I think that'll just about do it for me, and hopefully that's right on time. So thank you very much, everyone.
Thank you, Lachlan. I think I want to come and do a PhD with you.
No worries. Shoot me an email and we'll talk about it.
Okay.
Everyone else get in before she does.
Yeah. Okay, have you stopped sharing now? Okay, and our next presenter is Professor Frini Karianidis, who's from the School of Psychology. Thanks, Frini.
Thank you, Francis. Just checking that you can see my slides.
Yep.
Excellent, thank you. I'll put my timer on so I don't go over time. Okay, so I'm Frini Karianidis. I'm the Director of Functional Neuroimaging Lab here in the School of Psychology, and we're affiliated with the Stroke and Brain Injury Group and the Brain and Mental Health Program at HMRI, as well as with these scholarships are part of a package that is setting up Newcastle, Illinois Centre of Cognitive Neuroscience of Aging. So our primary collaborators on the projects that I'll present today are Monica Fabian and Gabriella Gratton, who have developed a methodology of looking at properties of regional properties of arterial health across the surface of the brain, and that give us insight into aging processes. Primary challenge we have, and what our group is trying to do, is to understand what it is that makes some people maintain healthy cognition into old age and other people show evidence of decline, even in the absence of any trauma or Alzheimer's related pathology, and to try and maintain people's cognition healthy for longer. So there are two projects for which we are recruiting. One is the Activate study that just started last week. We had a 6-month delay with COVID, but we're on the track now. And the key question there is to try and understand how lifestyle patterns of activity, nutrition and sleep affect cognitive and brain aging in older adults in the 60 to 70 year bracket. So this work is funded by an NHMRC Boosting Dementia Research Grant, and it is a collaboration between Newcastle and the University of South Australia, together with our researchers at the University of Illinois, Urbana-Champaign. We are recruiting both at Newcastle and Adelaide, and it's a 3-year study. It's a 5-year project, but a 3-year follow-up. So we're following people over 3 intervals, 18-month intervals, with the outcome goal being to develop a tool for older adults, an app that will allow people to tailor their best days. So look at what combination of activity, diet and sleep will produce the better predicted outcome of cognition, taking into account that any time you increase one type of activity, you have to reduce another type of activity, given that we still can't stretch a 24-hour day. So we have a range of different measurements that we're taking, both lifestyle, activity and nutrition analyses, neuropsychological and psychological assessments, as well as cognitive neuroscience measures that allow us to look at functional measures of cognitive control with EEG and functional MRI, structural measures of brain changes with age, but also these novel measures that use diffuse optical imaging to characterize regional variability in arterial elasticity and reactivity over the prefrontal cortex. The second study is what we call our ABC study. This is an ARC-funded project that we hope to start next year, which is basically trying to understand this link between arterial, brain and cognitive health in older adults again. And we're using an approach to try and understand this link where we will be implementing a cognitive manipulation, a cognitive intervention that is targeted to particular areas of the prefrontal cortex, and then looking at whether that intensive engagement of that area, of those areas, will improve arterial supply or the integrity of the arterial supply to those areas that are being engaged. So again, this is a collaboration with the Illinois group as well as Lumos Labs. You may have seen the Lumosity brain training apps out there. and with Monash and UNSW. And the outcome is to show that this intensive practice will change arterial supply and provide a protection against cognitive decline. And we're hoping to, we will be applying for further funding to do that follow-up study. Using a pre-post design and using also similar measures. I'm on time. Thank you, Francis.
Thank you, Frini. And you have how many scholarships going?
There are 4 available. They are domestic, and yeah, I've got a few people who are interested, but there are still opportunities.
Okay, thank you, Frini. Okay, our next presenter is Laureate Professor John Aitken, who's from the School of Biological Sciences. Thanks, Robert.
Okay, thank you, Francis. And so this is a research project which will be centered in the PRC for reproductive science. The reproductive sciences PRC is quite a large group of people. We have over 40 people here at Callaghan and more at the John Hunter Hospital. And we've had pretty much continuous funding from the ARC and the NHMRC for more than 20 years. And we focus very much on postgraduate research training. We currently have about 15 or 20 PhD students, and personally I've seen about 34 PhD students now through their degrees. We will provide, I think, quite a stimulating environment and supportive environment for anybody who wants to do a PhD with us. Now, the position itself is an interesting one. It's one which is funded by a biotech company, a Sydney-based biotech company called Memphasis. And it involves a collaboration between the University of Newcastle and a series of IVF clinics in Sydney under the auspices of Monash IVF. So this is a commercial industry university collaboration in the area of clinical embryology and assisted reproduction. And the project itself is basically trying to develop new methods for both diagnosing and treating infertility, human infertility. And we have a particular focus on the male. So we're very interested in trying to develop better diagnostic techniques for male infertility and improve ways of isolating sperm cells and encouraging them to fertilize oocytes. It's, I think, a unique opportunity to get to some leverage with a very important industry. And the industry itself is growing dramatically. We're seeing roughly a 10% increase per annum in the number of patients who are treated by IVF. We're seeing expansion of IVF clinics all over the world. And the qualification you would get, this PhD in reproductive health, would enable you to practice anywhere in the world. Clinical embryologists are very well remunerated, and this would provide you with a wonderful opportunity to engage with that industry. And indeed, many of my ex-PhD students are now running IVF programs in various parts of the world. So this is just a great opportunity to engage with the IVF industry and do some what I think is really intriguing research. These are very specialized cells that have a very interesting cell biology, and to use that to try to help people through some of the more difficult phases of their lives. Thank you.
John, if you can stop the share.
Yep.
Cool. Thank you very much. Our next presenter is Dr. Liam Phelan, who's talking about statistics for climate risk and uncertainty. Thanks, Liam.
Hey, thanks very much, Francis, for organising this session today. So we've got one scholarship that we're offering, and the discipline, if you like, for it is statistics, or perhaps applied mathematics, and it's very much in the area of climate change risk modelling, and also, and I guess looking particularly at uncertainty in that process. I guess that's the discipline it's in because we're wanting to take a more than disciplinary approach for this case study. So the core of it is in stats or applied maths, but the context is important as well. This scholarship is offered as an industry scholarship with an outfit called Climate Risk, and they're at the cutting edge of climate modelling as applied at very, very high resolution. So at the scale of postcode and even individual address. So thinking about what climate risks mean for very precise locations. So there's a lot of good work in climate modelling at global scale and regional scale and so on. When we start to apply that at a much higher resolution, then lots of different kinds of uncertainties come into the mix. So we can say with confidence, we can make claims with confidence about mean average temperature and so on at the end of the century at global scale, but thinking about what particular extreme weather events might mean for particular addresses, at the end of the century, even halfway through, is much more. There's a whole lot more work to do there, and there's so much uncertainty in that that really needs some exploring. Climate Risk does their modelling and is a commercial operation. So their clients include at least 2 of the big 4 banks in this country, and then also insurance firms in this country, and they're starting to work overseas as well. So we can think about climate risk for a particular building, a home, from the perspective of a homeowner, for example. And so homeowners are interested to know about, how their property may become increasingly vulnerable through time to, key climate risks. So in this country, that means things like coastal inundation, riverine flooding, bushfire, also drought through soil movement, leading to cracks in buildings and so on. So, and then of course wind as well. So there's other climate risks, but they're the key ones in this country. So for a homeowner, it's interesting to know about those risks, and that's important because home owners need to insure their properties, and insurance needs to be available, and part of availability means affordable. So insurance needs to be available and affordable. If we jump up a scale and think about the same question, perhaps from the perspective of a lender, then it's the same sorts of questions that are in play, but at a larger scale, because lenders are interested to be selling as many mortgages as they can, but they also need to know that their property portfolio is safe and secure. So it's interesting for lenders as well to know what's happening in terms of climate risk through to the end of the century and beyond. If I can just share my screen, I'm going to show here. This was a story on ABC Online late last year that was making use of climate risks work, and so the idea here is the rise of red zones of risk, if you like. So this is quite a good story, I think, in that it makes the work that they're doing accessible. I'm just going to flash through here. You won't be able to follow as I scroll through, but just to show what's happening here. Where is it now? So if we put in Newcastle, then we can get a sense that in the period through to 2100, we're going to see quite an increase in the percentage of addresses in that postcode area that will become potentially uninsurable. So it doesn't mean that they can't be insured in theory, they may still be insurable in that sense, but the insurance may be so expensive that it's unaffordable, and so it's not really accessible to folks. So it's a joint activity with us here at the uni and with Climate Risk. They're based in Newcastle. They're a small team, a tight team, and they're doing great work. I might leave it there, Francis.
Thank you, Liam. Okay, so our next presenter is Associate Professor Sally McFadden and Associate Professor Nathan Bartlett from the School of Psychology and somewhere else.
Oh, thanks, Francis. Look, Nathan is online somewhere. I'll just begin and then let him take over. I have 2 scholarships available. The first one is to look at. I'm not sure who is online here with us, but I'm looking for an engineer actually, or a physicist with interest in physics or biomedical device development. And so we've just got a new grant in, and the grant is to build some new novel technology for non-invasive imaging of the eye, and specifically we'll be trying to image functional properties of the eye non-invasively and the biomechanical properties of tissues within the eye. So that's the first project that we're looking for, basically either an optical physicist or an engineer of interest in biomedical technology and device development. The second project is with Nathan Bartlett, and I think he's online at the moment, so I'll let him take over. Hi, Nathan.
Hi Sally, how you going? Hi everybody.
Good, thanks.
I'll just quickly share my screen so we can all see what I'm talking about. Okay, so this is another PhD related to ocular-based research. This one really focuses on the clinical problem of ocular pain, which is the primary reason for visits to an eye care professional. And there are really no good drugs to treat ocular pain, which is caused by a variety of disease processes, damage, trauma, infection, etc. And so the objective of this PhD proposal is to really work with industry and to develop new drugs that target ocular pain, particularly for corneal or topical delivery, in order to really deal with the pain, but also to help ameliorate some of the disease processes that underpin that ocular pain. Now, this really is a unique opportunity. So this is an industry-engaged PhD scholarship, which is jointly funded by the New South Wales Ministry of Health. I'm in partnership with industry. And so the industry partners are 4A Therapeutics, which are funded by venture capital, Brand and Capital Partners. And they're collaborating with the CSIRO synthetic biology unit down in Melbourne. And they're working together to develop a novel drug delivery platform, and which will be focused at delivering medications that prevent or reduce ocular pain, but also help with repairing the disease processes that underpin that. So it's a big PhD. It's going to require a very special candidate, because you'll be spending approximately 2/3 of your time at labs with me and with Sally, working on developing human corneal epithelial disease models. My lab has extensive experience in developing epithelial models, mostly in the airways. Now we're working on developing corneal models as well. And that'll really be supported with expertise from Sally, who has, as you know, extensive expertise in eye research. And then you'll be spending some time down in the CSIRO synthetic biology labs in Melbourne, actually synthesizing the new drugs, compounds that you'll be then coming back to uni Newcastle to test. And I say all travel and living expenses are paid for during your time down in Melbourne. But it's also going to be engagement with industry and venture capital. So there's going to be a very large focus on understanding translation and commercialization of drugs, of new medications. And so time will be spent at that venture capital office, Brand and Capital Partners, either in Sydney or Melbourne. And you'll have a chance then to engage with investors, talk to them, learn about what life is like in biotech commercialization. You'll get to attend the Oztech Biotech Conference. And again, all expenses, travel and attendance at conference, etc., will be paid for as part of this You'll also notice a very generous stipend, so it's 40K per year for the first 3 years, and then it reverts back to the more standard stipend after that. So again, I guess encouraging you to think like a commercial, industry-focused researcher in getting things done, hitting your milestones, getting the research done. I should also say that the IP is owned by Foray, but there will certainly be opportunity for inventorship on patents if you play a key role, which you would be expected to in the development of these new drug platforms that would then progress onto clinical development. So I'll stop sharing and just to reiterate that this is a very unique PhD opportunity. As I said, it will involve some movement around and multiple exposures and engagements in different spaces around both basic research, drug development and commercialization. So it's going to, I say, take a very special kind of PhD candidates, someone with interests in chemistry, but also in cell biology, but also then interests in industry engagement and commercialization. So I'll leave it there. Thanks.
Thank you, Nathan. You have nothing further, Sally?
No, that's it, unless we've got more time. I doubt it.
No, you have no more time, sorry, but I wouldn't have allowed a sentence or two. Okay, so thank you very much, Sally and Nathan. And our next presenter is Professor Brett Nealon, who is the Global Innovation Chair in Biotechnology. Thanks, Brett.
Thank you, Francis. I hope you can hear me and it's all okay with the screen.
Yes, it's fine. You might want to make it go the whole screen because you've got all the slides up on the left-hand side. If you go down the bottom right, yeah, that one.
Yeah, sorry about the background noise too. We're renovating something outside again. So my laboratory really looks at, or starts with looking at various environments around our planet. We call them extreme environments because they're not where humans tend to live. But we look for the microbes in those environments, and we really look for the physiologies of those microbes to see what type of valuable natural products can be sourced from them. A lot of times these microbes can't be grown in the laboratory, so what we do is we start with the genetics of those microbes, extract the DNA from these extreme environments, and then clone those genes and then produce the products that way. So in a way, it's synthetic biology or genetic engineering to access the biodiversity or the genetic diversity of Earth. And that's just information where you can contact us if you wish. I'm just going to give you a few examples of where we've been doing work. One of these is in the coastal regions of Western Australia, and I don't know, some of our work really crosses over between disciplines. While I'm a biologist, we work with geologists to look at how these rocks were formed early on in the evolution of Earth or evolution of life on Earth. And the interesting thing about here, the biotechnology that we find here, is that there's novel antibiotics being produced by these bacteria. But also living in this environment, they produce UV blocking substances, which are now becoming replacements for sunscreens, and also components in new materials that you can use in space travel, for example, where there's high UV environments. The second project that we work on, one of the other projects we work on, is using Indigenous knowledge to understand why certain plants have been used for medicines for tens of thousands of years. And what turns out to be interesting about this project is it really crosses over with a bit of the social sciences and also plant biology, because the bacteria and the fungi that we work on actually live inside these medicinal plants. And again, these become sources of new drugs. We do this in collaboration with the various Indigenous people of Australia and China and Indonesia. And then once we find these genes or these pieces of DNA, we clone them. And then what we've got to do is produce those compounds in large enough amounts for them to be useful for industry. And this is where, like, the scale up of the biotechnology becomes important, And we have a new Centre of Excellence for Synthetic Biology, which really is about how do we use waste, for example, to be the feedstock for these production of these factories to produce drugs and other valuable compounds like sunscreens that I mentioned. The other PhD project that we have available is more pure environmental microbiology, and that's looking at water quality and also the tracking of antimicrobial resistance in wastewater. So this project is really microbiology, but it does have a lot of crossover with bioinformatics. So large data set type genomic work is how we analyse the bacterial populations in these impacted systems. So the skills we're looking for are, basically third year or fourth year honours graduates in microbiology, biochemistry, genetics, molecular biology, all those types of sciences. More recently, people with mathematics background, with interests in biology. So that's where I've got basic statistics. The training we offer is in bioinformatics, mathematics and mathematical biology, and then more advanced omics, including genomics and proteomics. We do a lot of advanced molecular cloning, especially of large genes, and the expression of those genes and the scale up of production of the compounds that we're of interest. We also, at the tail end of a lot of our work, do a fair bit of preparative and analytical chemistry. So you can see we're going from environmental type microbiology through bioinformatics right through to chemistry. And this is all pretty standard, as you've heard before. The interesting thing that you guys who are attempting to do a PhD should realise that part of your PhD will be within the labs here at university. It could be at labs in other universities, but it also could be within the industries. And as you've heard just recently, a lot of us work with various industries that could potentially be your employers, sorry. And also that the labs here, we have a lot of internal support in terms of equipment, but there's also a lot of other, say the postdoctoral scientists, other PhDs in your laboratory that you really form a cohort of people who are doing research. It's a very big change from, say, the type of training you get as an undergrad or even an honours student to what you'll end up doing as a PhD student. So that's all I've got to say. Thank you.
Thanks, Brett. And how many scholarships do you have available?
That's two Francis.
Cool. Thanks. Okay, our next presenter is Professor Jenny Bowman from the School of Psychology. Thanks, Jenny.
Thanks, Francis. Just trying to share my screen. How's that? Visible?
It's not yet, but it's coming, I'm guessing. Yes.
Beautiful.
But you might want to click that whole screen thing down the bottom right.
Where is it?
Yep.
There you go. Thanks for the opportunity to present this afternoon, Francis. So I'm in psychology. My particular area of interest in psychology is health psychology. And in particular, now for probably the last 15 years or so, it's really been focused on improving the physical well-being of people who are living with a mental health condition. So initially, my interest was in chronic disease prevention generally. At a population level, what can we do to help prevent chronic diseases such as cardiovascular disease, cancers, diabetes, which are the major causes of morbidity and mortality for us as a general population. However, I came to realise that within the general population, there are many subgroups who experience a great deal of inequity in terms of that chronic disease burden. And one group who's particularly notable in that respect are those people who are living with a mental health condition. And it's fairly stunning figure that many of us don't realise, but that the life expectancy of people who live with a mental health condition is really substantively less than for other Australians. So 12 to 16 years, up to 20 years loss of life, depending on the particular severity of mental illness that someone is experiencing. Most of that reduced longevity is not due to things such as suicide, although some of it is, but in fact the majority of that loss of life is really due to those same chronic diseases. So people with a mental health condition are more likely to develop and then to die from those same physical chronic diseases as the rest of the population. Working in chronic disease, the upside is that much of it is potentially preventable. So if we can in fact help people to make change to some significant health risk behaviours that increase the risk of chronic disease, then we can actually reduce that risk significantly. So those risk factors, we often use the acronym SNAP: smoking, nutrition, harmful consumption of alcohol, and inadequate physical activity. And sleep is, I guess, gaining prominence as well as a risk factor for chronic disease. So for people with a mental health condition, we need to do a better job of providing that population with access to evidence-based care to help support people to reduce those modifiable health risk behaviours should they choose to do that. We know that despite policy and guidelines that preventive care and behaviour change support should be being provided to people in mental health services, it is very often not. And so a significant opportunity to help reduce the chronic disease burden isn't being realised. So most of our research actually involves working with health services and other agencies that have contact already with people living with mental health conditions and provide various kinds of support to those people. We work with health services and other agencies to reorient the care they provide to include preventive health care for health risk behaviours. So most recently, we've been awarded a grant from the NHMRC, from the Medical Research Fund, to develop and test a new model of providing preventive care within government community mental health services. So these are services that provide really the majority of outpatient mental health care in our communities. So the opportunity to reach many people is large. So the model of care that we're testing is a dedicated provider who will integrate within community mental health teams to undertake an initial assessment and to start to develop a plan with mental health clients around behaviour change. It's also a project that involves increasing the capacity of mental health clinicians in the services so that they can continue on working with consumers to help behaviour change happen. It's going to be undertaken as a cluster randomised controlled trial across 3 New South Wales LHDs, Hunter New England, Central Coast and Mid-North Coast in New South Wales. And it's going to involve assessing both the effectiveness and the cost effectiveness of this intervention. Delivering preventive care to clients will be the primary outcome of interest. So within that grant, we have 2 PhD scholarships funded. We're keen to, for those to go to excellent PhD students. We're also, of course, always very keen to talk to people wanting to do PhDs. So not necessarily just with this project, but across the range of work that we do. So yeah, please get in touch. If any of that sounds at all interesting, we'd love to talk to you. Thanks.
Thanks, Jenny. Cool.
Thanks, Frances.
Okay. Our next presenter is Dr. Diane Rayboursay. Sorry, I can't
That's
pronounce the name.
That's fine, Rosia.
Who's from the School of Biological Sciences. Thank you, Diane.
Thank you for giving me this opportunity to present first the lab and a potential project within the group. So I'm a researcher in the Smith group, and what I'll do is I'll just introduce the team first and then the project. So the core of the group arrived from Edinburgh, Scotland to Newcastle to follow Lee Smith, who joined as the PVC. And the first task that we had with Anne-Louise Gannon, who will be presenting a project later on, was to develop a fully functional lab, which is also fully funded as well. So here's the fancy new lab that we have. And so the group is composed of a multidisciplinary team, which have different biological background as well, but an interest mainly in endocrinology and the improvement of male health. So we have an interest as well in steroids, more specifically androgens. We know that androgens are crucial for the masculinization of males, development of their reproductive system, but not only. They've been shown to be crucial for men's health and well-being. And there is an accumulation of evidence showing that low androgens are actually associated to the development of metabolic disorder, such as obesity, cardiovascular disease, and mental health issue, as an example. So the group aim is to understand how androgens and steroid production are regulated in order to develop specific and targeted therapy. So, as mentioned above, we are interested in understanding the regulation of the endocrine function of the testis, but also the adrenal, which is another organ that is steroid productive, and in order to promote or improve lifelong health. And we want to understand better the system to prevent or treat disease, infertility as well, and identify new targets for therapy. And the group has a specific interest as well in developing novel approaches to understand and develop new therapies. And this involves the use of fields such as biotechnology and development of nanotools, viral vector as a tool to deliver gene therapy. And we do use a lot of transgenic in vitro and in vivo model. And those approaches are also associated to cutting-edge omics, involving bioinformatic and computational modeling. And the aim overall is really to better grasp the mechanism and regulation underpinning endocrine regulation, but also involved in endocrine-related disorder. One of the projects that we have in the lab is focusing specifically on understanding the interaction and crosstalk between the adrenal and the testis. And under stress condition or illness, it's been shown that we have an impact on the steroid production, and people tend to show low testosterone production. And we know that stress and illness are mediated by, impact is mediated by glucocorticoids, which are secreted by the adrenal glands. The mechanism involved in that regulation are still unclear. And we know that under stress, fertility becomes a second consideration upon survival. So the aim of the project is to better understand the regulation involved in that system, with a focus on the receptor on which the glucocorticoids have an impact, which is the glucocorticoid receptor, and look at the impact on testosterone production. The aim being that by understanding how the system is regulated, we'll have a better depiction of the genes involved, which means that we can then potentially develop a new gene therapy. So this model and this project will combine tools that we commonly use in the lab, such as in vitro and in vivo models, as mentioned earlier, as well as in silico modeling. So that will give us a sort of nice overview of the regulation, and also using our biotechnology developed in the lab. This project will also be supervised by Dr. Anne Lewis-Garnon and Professor Lee Smith. Thank you for your attention, and please feel free to get in touch with myself and to have a look at the Swiss Lab website. Thank you very much.
Thank you, Diane. And how many scholarships are available for that project?
There are 3 scholarships available in the group, and more projects as well, but we're just presenting this one, and Anne will present another one as well.
Okay, thank you.
Thank you.
Okay, our next presenter is Associate Professor Bill Leggett from Marine Science.
Thanks, Francis. Thanks for coming. So as Francis, I'm a professor down at Arimba, and my research group focuses on corals and the implications of climate change on corals and anthropogenic stressors. So my group spans a broad array of disciplines. So we have projects looking at microbial symbioses, gene sequencing, metabolomics, all the way through to physiology and even ecology. So coral reefs around the world support anywhere between about 500 and 1 billion people. And they're currently under threat from climate change. So particular increasing temperatures leads to a phenomenon called coral bleaching, where corals go from sort of color here to this really pale color here because they lose their symbiosis. And if these coral bleaching events continue long enough, you'll actually end up with the loss of corals. So we had global coral bleaching events from 2014 to 2017. And over the last 3 years, we've lost somewhere in the order of about 30 to 40% of our corals from the Great Barrier Reef. And in the future, this is really only going to increase as we increase in temperature. We have a PhD scholarship that's associated with ARC Discovery that we've just received. It's actually a scholarship through the UTS, but can be based at either UTS or Arimba. And it's to work with researchers here at UN, researchers at UTS, and also a postdoc who's been employed with the project, and also Oregon University in the US has traveled to the US here. And the project is based around investigating what we call biogenic volatile compounds. So compounds that are produced by corals. And these can have a number of really interesting implications in terms of signaling between corals, signaling between other organisms, and even some compounds that corals produce can actually change the weather in terms of making a compound called DMSP. So we've got one scholarship, but we've really got 2 projects depending upon what is of interest to the applicant. So we have one project which is to develop what we call first metabolic models for the biogenic volatile compounds. So this is very much more a molecular project, which would really be suitable for students with more of a molecular biochemistry background. So the really main questions around this are to looking at seeing how these compounds that are produced by corals change under stress, and then looking at particular gene pathways and metabolic pathways that are involved in its production. So as I said, it would really involve gene sequencing, bioinformatics, and also metabolomics. So we've got expertise in all of those. And then looking at how these different pathways are influenced by different taxa. So are particular taxa, coral taxa, producing these differently? The second project that a student could undertake is a bit more physiological and ecological. So it's looking at biogenic volatile organic compounds, BVOCs, but looking at how they're actually involved in environmental cues and communication between organisms. So thinking about how corals can actually communicate with these compounds to either other corals or other organisms in the ocean, in the marine systems. So if you undertook either of these, you'd be looking at getting expertise in coral stress experiments. Obviously, there aren't many corals down here, so field work would be generally conducted at Heron Island. We also conduct field work at Lord Howe Island, Hawaii, and I've also got some students who are currently in the Pacific or will be in the Pacific when COVID clears up at Samoa. So there's a variety of travel opportunities there. And you'd be looking at applying developing state-of-the-art biochemical and molecular methods and applying these sort of bioinformatic techniques. So it's really a cross-disciplinary project. Only requirements we have, unfortunately, you have to be an Australian or New Zealand citizen, and familiarity with marine systems, in particular corals, is really not essential. It'd be nice if you have it, but because of the nature of these projects, where it is very much more molecular focused, we're quite happy to take on students that really have a strong molecular biochemical background. And we can bring them up to speed on more of the ecological marine system. If you've got any interest in that, please give me contact. And there's my email there, Bill Leggett. Thanks for that, Francis.
Okay, thanks, Bill. Okay, our next presenter is Associate Professor Jeff Hogan from the School of Mathematics. Thanks, Jeff.
Thanks, Francis. Does that look okay?
Yep.
Okay, so yeah, thanks. The one scholarship we have is for is associated with an IRC discovery project, and it's for a PhD in maths, although the project will require theoretical and laboratory work, and I'll just describe where the work is exactly. So imagine you have a big sample of, for example, iron ore, and you want to work out the mass distribution of the particles as a function of the density. So this is definitely an important aim when you're working with, let's say, iron ore. And the sink float method is often used, which uses a series of baths with liquids of different densities. And what that means is that you've got this this bath with, let's say, water in it, and you feed the ore in at the top, and some will sink and some will float, and the stuff that floats is very light, and you skim that off, and then you add a liquid of higher density, and then you get some more stuff floating to the top, and then by the time you get the really nasty stuff, the heavy stuff, the liquids that you're using are really quite nasty. They are often quite toxic. So you want to reproduce the so-called washability data, which is gives you the distribution of densities and masses across the, as in size rather, across the sample, but you don't want to use the nasty liquids. So Kevin Galvin, who will be the other supervisor of this PhD, and has developed something called the reflux classifier, which is this big machine of, as you can see on the left, inclined plates. And you put the ore in the top, and it sits in the inclined plates, and then you force, let's say, water in from the bottom. And over on the right, you'll see that there's this hydrodynamic process going on, which lifts some of the material out, and it will lift, of course, more out the higher the pressure of the water or whatever other liquid you're using. And so you get these series of data which you want to so-called deconvolve. So the hydrodynamic forces are optimized and controlled, and unfortunately, there's this hydrodynamic dispersion that goes on, and so there's a convolution or a mixing of the overflow fractions. And the problem, of course, is to determine the true density distribution. And what you have is the mathematical model is this convolution model that I've written up here for the mathematicians. You're given the GIs, and what are they? They are the amounts of material that come out from various velocities of fluid forced into the reflux classifier. And K is a kernel that does the mixing up of the various densities and masses. And you get a different kernel for every different velocity, V sub I. And so what's required here is that we learn the fractionation kernel K. And then given that we know the kernel, we want to be able to deconvolve the fractionation data. So given the GIs on the left, work out what W is essentially. And the solution from the analysis that we've done so far looks like it will require techniques from optimization and harmonic analysis, particularly what are called frames and wavelets, which is my specialty, and integral equations. And the applicant we're looking for will have a good record in mathematics, honours in mathematics or related disciplines. Someone in, let's say, electrical engineering would be good too. And this will be a theoretical and laboratory based PhD, as this is a very cross-disciplinary project. And we're looking for someone in expertise in at least one of harmonic analysis, optimization, numerical analysis, or signal processing would be great. And programming skills in MATLAB or Python would be desirable. And that's all I have to say. If anyone has any questions, I'd be delighted to accept emails. So thanks, Francis.
Thanks, Jeff. Okay, our next presenter is Dr. Anne-Louise Gannon from the School of Biological Sciences. Thank you, Anne-Louise.
Hi, thanks, Francis. I'm just going to share my screen.
Yep, all good.
Cool. So I'm very excited to be speaking to you today about a scholarship that we have available in our lab. So you've already heard from my colleague, Dr. Diane Raborse, on one of our scholarships in the Testis. So I'm here to speak to you today about one of the scholarships that we have that's more focused on the adrenal. So the title for this project is Targeting Pharmaceuticals and Nanoparticle Gene Therapy Vectors to Treat Adrenal Pathology. So if you stick that into the university's website, you'll also be able to find a more in-depth description of this PhD scholarship if you're interested. So this PhD scholarship is going to be supervised by myself, Dr. Anne-Louise Gannon, and Professor Lee Smith. So if you're interested in contacting us, our emails are here. It'll also be at the end of the presentation as well. So just to give you a little bit of introduction to the lab, in addition to what Deanne has said, so we are interested, as she's described, in the endocrine system, and that encapsulates a wide array of organs. But where our expertise lies within the pituitary, the adrenal, and the testis, and the interplay between these organs. So my area of expertise is in the adrenal, and this PhD scholarship is going to focus on adrenal biology and repairing adrenal biology. So just to introduce that a little bit more, to give you a bit of context of the importance of the adrenal. The adrenal is found on top of the kidney, sits there like a little bit party hat, and it's responsible for producing some very key steroids and hormones. So it does this from very specialized zones. The medulla, which is responsible for our fight and flight response, which people are probably most common with, but where we're very interested in is the adrenal cortex, which produces the steroids we're interested in. So some of these steroids are mineralocorticoids, which is responsible for our salt balance, so things like blood pressure. Sugars in the form of glucocorticoids, which is responsible for regulating our stress response, and also sex steroids that are important for the development of secondary sexual characteristics. So as I'm sure you can imagine, that an organ that produces such a wide array of hormones and hormones that impact nearly every body system, it is imperative that these are maintained and these stay producing as normal throughout someone's life course, and that's for male and females. So what current problems do we have in adrenal research that we hope to begin starting to address with this PhD project? So adrenal regulation, so these very specified and specialized zones within the adrenal and how they produce the steroids is still a massive unknown. And because we don't really understand adrenal regulation, this means that adrenal disease is fairly prevalent, and we don't really have many treatments to treat any conditions that results in adrenal deficiencies. And these can range from depression, Cushing's syndrome, Addison's disease, and of particular focus for this PhD scholarship is congenital adrenal hyperplasia. Now, what happens in patients that have congenital adrenal hyperplasia is that they have a deficiency in one of the enzymes that is crucial for the production of all three key steroids from the adrenal. It results in the loss of mineralocorticoids and glucocorticoids and massive excess of adrenal androgens. The current treatment regime for these patients is lifelong steroid administration, which is not only very difficult to manage and can be life-threatening if it is not managed correctly, but also very expensive depending on what healthcare system you're with. And part of the reason that we don't understand adrenal regulation and adrenal disease is because relevant mouse models are severely lacking. And this is part in due because the adrenal, mouse adrenal and the human adrenal aren't genetically identical. So this has impacts for other diseases, studying other diseases such as prostate cancer, polycystic ovarian syndrome, and the role of adrenal androgens in the development of those secondary sexual characteristics. So we're, as a team, very interested in nanobiotechnology, especially in using these as potential treatments and therapies. Now, nanobiotechnology has been used in numerous contexts in medicine, and where we're particularly focused on, we'll be using these nanotechnologies in the form of therapeutics and regenerative medicine. So how can we go about using these nanomaterials to address these problems, and what sort of technology will we be using throughout the course of this PhD? So at the minute, we've currently developed nanoparticles in the laboratory that can carry a range of different cargoes. And of particular interest is these nanoparticles are able to be specifically targeted to carry these different types of cargo directly to the adrenal cortex whilst leaving the rest of the body untouched. So this is important for drug delivery in the instances where we have adrenal excess, steroid excess. To develop on that further, we also have the ability to deliver viral and gene vectors that should hopefully repair that genetic deficiency that's causing the problem to begin with. And if we're able to manipulate the adrenal and its genetics, we'll also be able to develop more relevant and human relevant mouse models. So skills developed as part of this PhD, you'll develop an expertise in the field of endocrinology. Design, and you'll utilize a range of nanobiotechnology from viral vectors down to just normal plasmids. You'll be fully trained in mouse model generation, animal handling and husbandry, as well as cell culture and utilizing computational and biological modeling and bioinformatics. So you'll come out of this PhD with an incredibly amazing skill set from in silico, in vitro and in vivo technologies, which will put you incredibly well placed, not only in the field of endocrinology, but for anything that you particularly want to do after your PhD. And if we are ever able to leave the country again, we also have an established lab in the University of Edinburgh, where part of our team originally moved from. So you not only have the opportunity to be trained and gain skills at the University of Newcastle, but also a world-leading institution at University of Edinburgh in Scotland as well. I'd just like to say thank you for giving me the opportunity to speak to you today. If you're interested in knowing a little bit about me and the team, you can have a look at our website here and our team's website. And I'd just like to finish off by saying thank you very much.
Thank you, Anne-Louise. And that's the same 3 scholarships as Diane?
It is indeed, yes.
Yeah. Okay. Right, so 3 scholarships in that group. Thanks. Our next presenter is Dr. Hannah Schunker and Associate Professor David Ponton from Physics. Thanks, Hannah and David.
Thank you. After you.
All right, hopefully everyone can see our slides. So I'm David, this is Hannah. We work in solar physics, and so some of the sorts of things that we are interested in explaining are the likes of this eruption that you see in the atmosphere of the sun. We're looking at...
we don't see it. We don't see any slides. You haven't shared the screen.
Try again. And choose desktop 1 and share.
Okay. Now we can see it.
Excellent.
Thank you.
Okay. So now can you see one of our spectacular eruptions on the sun? Okay, hope you're seeing that now. So this is an eruption of billions of tons of plasma out of the sun's atmosphere as one of the many dynamic phenomena that we see on the sun that we don't really understand. And one of the motivations to understand these is that these explosions generate enormous bubbles of hot plasma that get shot out across the solar system, sometimes heading towards the Earth. Now, those explosions are driven by magnetic forces in the sun's atmosphere in ways that we don't understand. And there are also lots of other dynamic phenomena that are associated with the sun's magnetic fields that we are seeking to understand in our group.
So these huge explosions that eject charged particles into the solar wind, and if they're directed towards Earth, they can impact the satellites, communications, aviation, power, etc., and they can disturb these systems. And as human society is becoming more and more reliant on technology, this is becoming more and more important to understand the physics behind it so that we can be more successful in forecasting space weather. So all of this is driven by the magnetic field of the sun, as David mentioned, everything you saw there, all that activity, those bright features that are very dynamic, that's all the sun's magnetic field. And the magnetic field originates in the interior somewhere, and we don't know where. Once we can understand that, we will have a better idea of how to predict what's happening up through the surface and out into the solar wind. So these are two really fundamental problems in astrophysics, is understanding these eruptions, how the magnetic field reconnects and how it's dynamic in the atmosphere, as well as where the internal magnetic field is inside the sun and stars. So our ideal student doesn't necessarily need to have a background in physics, but something along the lines of physics, mathematics, or some kind of computer science field. Of course, we would like them to be extremely brilliant, but it's very important for us that you have good scientific communication skills, easy to talk to, strong coding skills, and we want you to be motivated to understand the physics problem. So to really get into the science of it and to solve some problems. The skills we think you will leave with is quite a generic one. If you're doing a PhD, is independent problem solving. You will solve your own problems and you will guide your own project. The second one we can offer is experience with big data. There is just so much data in astrophysics and solar physics these days. This requires efficient computational analysis. The other thing we can offer is high performance computing experience, running numerical, three-dimensional numerical MHD simulations.
So to find out more, come and talk to us. And these are our email addresses up down here. So just send us an email. I guess one last thing to plug in is that you know, it's a great time to be studying space in general. The space industry is really taking off. The space industry, particularly in Australia, is taking off, and space weather is really important to society. So to do something that is relevant and also fun, then yeah, try solar physics.
Okay. Thank you.
And thank you, Hannah. Thank you, David. Cool. And our last presenter in this series is Laureate Professor George Willis, who's from the School of Mathematics. Thanks, George.
Okay, thank you, Frances. I'm going to talk about possible projects to do with, in mathematics, to do with the symmetry of networks. This is basic research in mathematics. Now, I don't have slides. I'm going to try to present something on the iPad. And so I'm going to share the screen on the iPad. There's an analogy between what we're, oh, sorry. I need to share screen before I do that. Okay. Can you all see my a screen now, a blank screen?
We can see a blank screen, fine.
Okay, good. So there's an analogy between the research I'm doing and eigenvalues, and I want to just say something briefly about that, just to illustrate the ideas. So here we have our usual horizontal and vertical axes, and we imagine acting on this space by multiplying by some 2 by 2 matrix. What this does is if r is less than 0, it shrinks in the horizontal or x direction by a factor of r, and then expands in the vertical direction by a factor of 1 over r. And the dynamics then, if we look at a general point, it follows a hyperbolic curve, like so. Now, this is what might be thought of as smooth or continuous dynamics or symmetry, and this is very well understood. Has been understood. The mathematics of this has been understood for a lot longer than a century, and it has been applied in areas such as mathematical physics and quantum mechanics and relativity. Now, as I said, this is well understood, and this is not what the research is about. The research is about symmetry of networks. And now I want to go to another screen. So here, if you can see a picture now, this is meant to be a picture of a network, an example of what's called a regular tree. So here we have vertices or nodes, and every one has 5 neighbors. And although I can't draw it, you should imagine this is continuing indefinitely, so every vertex has 5 neighbors. Now, this is a particularly simple example of the sort of network that we are interested in, or the symmetry of these networks. Now, the symmetry is not continuous or smooth in this case, because if you imagine the symmetry which sort of rotates about the central vertex shown there, to take one node to another node, one of its neighbors to another neighbor by rotating through 1/5 of a rotation, for example, this really, it can't be done smoothly without sort of leaving the network. To jump from one node to another and stay within the network, it really has to jump. It can't be a smooth transition. And that's the sort of symmetry that we're looking at here, the symmetry of networks where the transitions have to be discrete or have some jumps. They cannot be smooth transitions, as in the case of the dynamics, which I showed you earlier. Oh, I see we just increased the number of levels of the tree, but yeah, it gets pretty hard to draw as the number of levels increases. I guess this is indicating some connection with the idea of fractals, which some of you might have heard of. There is a connection between fractals and what we're doing. But yeah, so the discovery of smooth symmetry, the smooth symmetry has been understood for more than a century, but a recent discovery is that in the symmetry of networks, there is an analogous idea to eigenvalues. And now, to illustrate this, we draw the same tree as I drew earlier, but we draw it in a different way. And now you can see that a symmetry of this would be a translation. The previous one, the obvious symmetry was a rotation. For this, if we draw it this way, the obvious symmetry now is a translation. And the discovery is that from recent times is that if we have a general network, there is always, and it might not be such a simple example as a tree, it could be a very complicated network, but there's always some analogous structure corresponding to this translation of a tree. So the trees emerge sort of naturally in general symmetric structures in the same way as we find the eigenvalues of a matrix. So that's the relatively recent discovery, and the project now, and this is part of an ARC-funded project on what we're calling zero-dimensional symmetry. And the project now is to try to understand this symmetry and classify the symmetry and understand the general algebraic structure. We're trying to get an understanding that would be suitable for making computer algebra packages to deal with the sort of mathematics that we're working with. There are no really such algebra packages, computer algebra packages at the moment. Now, the assumed knowledge is, well, algebra and group theory in particular. And I'd also want some notion of some understanding of topology, because these are infinite structures. We cannot compute with them precisely. We have to work by approximations. And that will be part of the what we're trying to understand, how we do approximations to the computations in these structures. Now, you'll be working as part of a research group, and the other members are shown on this page here. And if you go to this this page, you can find out more about our activities. So just enter zero-dimensional.group into Google, and it'll take you to this page essentially. And there you'll see some of the other team members and some of our activities, including, if you look under the miscellaneous tab, there's the tool that we use to draw these pictures. So this is these sort of visualization and computational tools are things that we are developing as we go. Yeah, I think I'll stop there. Thank you.
Thank you, George.
Stop broadcast.
Yes, well done. Okay. That was the last of our presentations of people who have scholarships. Remember that the best plan now is to think about what people have told you. Think about whether one of the areas that people talk to you about is interesting to you. Check whether you've got at least some background in that area or none if you're interested in Bill Leggett's study. And then email that particular person and make a meeting to have a chat to them. Your program here has links to all the sites for all the presenters, and you'll be able to find their emails just by clicking on those links. Okay, are there any questions? We've got about 5 minutes that we could have any questions if anybody has any. If you could, if you could put them up in the chat. Nothing in the chat. Where's the chat? Oh, here it is. Yeah. Okay, well, thank you very much to all our, what is the cut-off date for applications for these scholarships? There is no cut-off date for these scholarships. They're being held by the researchers who've been talking to you, and they will tell you when they would like you to apply for it when you talk to them. It's not like the normal main round that the university runs where the cut-off date was the 1st of September. It's just go and talk to the person, see if they think that you'd be a good mix for their particular project that they have the scholarship for. Okay, any other quick questions? Okay, well, thank you very much, everybody, for your presentations. I enjoyed listening to them. There are at least 5 PhDs that I would like to do now, having already gotten one of all the talk, not the maths ones, George, sorry. But some of the other ones looked really, really interesting, and I'm sure there are maths students out there who found the network symmetry stuff fascinating and would like to come and do a PhD with you. Okay, and thank you to all of you people who attended. I hope that some of you develop a good relationship with some of the supervisors and end up getting a scholarship and doing your PhD in that area. And we'll finish now. Thank you, everyone.